Share the screen. So today we're going to be finishing chapter 2 and starting chapter 3. In the lab today, we will be doing lab 3. And don't forget, you need to start the infectious disease project. I have not checked my email to see if anyone has emailed me, but I will check it after class. Any questions? If not, let's begin with chapter 2. Now, why did that close? So we had finished talking about carbohydrates, is that correct? And we need to talk about lipids. I'm not hearing anyone say anything, so I'm going to assume this is correct.
Yes.
Thank you. So lipids consist of carbon, hydrogen, and oxygen, but they don't have much oxygen. Lipids typically are nonpolar and insoluble in water. We call them hydrophobic. They don't dissolve in water. There is one exception, but all the other lipids are hydrophobic. Lipids are structural and energy storage roles, or they have storage roles in cells. Lipids are primarily the components of cell membranes. And as you know, if you eat too much food, it'll be stored down as lipids, and we call that fat. Any question about any of that? There are 3 types of lipids: a simple lipid, complex lipids, and steroids. Let's talk about each of these 3 types of lipids. Let me go ahead and erase that. Before I move. Okay, of the simple lipids, they can be made of fatty acids, and they are a major component of most lipids. Not all, but most lipids. They consist of chains of carbon, and they're long chains of mostly hydrocarbon. And here we're seeing one. Let me see if I can blow this up. Right here. So long chains of carbon with hydrogen bound to the carbon. The carbon is the backbone. And there is a little bit of oxygen in this COH carboxyl group. Saturated fatty acids have single carbon bonds, where one carbon is linked to another carbon with a single covalent bond, and then all the other links have hydrogen. An unsaturated fatty acid has one or more double carbon bond, which you'll have, come on mouse, right here you'd have 2 carbon covalent bonds. And carbon can only bind to 4 things, so it'll have 2 less hydrogen, so that hydrogen won't be there, and that hydrogen won't be there. And then this carbon will bond once here, once there, and then 2 times to carbon. Everybody got that? So that's an unsaturated fatty acid, and where you have the double carbon bond, it'll cause a kink in this chain, which is shown in the next slide. Let me blow that one up too. Move this over here. Oops, didn't mean to do that. So where you have the double carbon bond, which you can see here, there's less hydrogen on these two carbons because they're double bonded than, let's say, those two carbons, which are single bonded. And where there's the double carbon bond, there's a kink in the chain. So that's an unsaturated fatty acid, and unsaturated fatty acids have at least one double carbon bond. A saturated fatty acid are all single carbon bonds. When we're talking about simple lipids, we call them fats or triglycerides. They do contain fatty acid moieties and glycerol. Glycerol shown here, and then a fatty acid moiety here. And they're bound together in a dehydration synthesis, meaning water comes out, the hydrogen from the glycerol and the OH from the carboxyl end of the fatty acid. In a triglyceride or a fat, you have three fatty acids bound to the glycerol, and that's why we call it a triglyceride. There are three fatty acids bound to the glycerol. Triglycerides are the most common source of fat in animals. And if you want to see some triglycerides, just look down at your belly. They are the body's richest energy source. So if you need energy, and let's say you're in a frozen environment and you have to walk a long distance and you got to carry all the food, and you're going to need to eat along the way because, well, for one thing, it's freezing, and for the second, you need the energy to walk there, and you're saying, what should I take? You should take fats because they are have the highest energy source. I'm not saying they're going to be the healthiest food for you. I'm saying they're going to give you the most energy. Any question about any of that? End the show here. Now, why did I go backwards, I think? Yep, I did. I went backwards. Let's go forward. All right. So triglycerides are the most common fat in your body, and they're also the most common fat in the diet. The key is that you want to get some unsaturated fatty acids in your fats so that you can eat unsaturated fats, which are healthier than eating fats. Lipids are also important in this. Sorry. Lipids are also important in the cell membrane. And in the cell membrane, we have a special lipid that we call a phospholipid. And the phospholipid is the main molecule in your cell membranes. A phospholipid is a molecule that does have two fatty acids, and one is always saturated, the other is always unsaturated, and then it's linked together on glycerol. So there's glycerol right there. And the difference is in the third group of the glycerol, we have a phosphate group attached to the phospholipid, and that's why it's called the phospholipid. Phospholipids are unusual in that they are a lipid which can dissolve in water. They are also a lipid which can dissolve in oil, and that makes them biphasic. there are not too many substances which are biphasic, but you do know of some, and that is soaps and detergents are biphasic. And the reason why soap washes your clothes better is because the soap can dissolve in the water and then help take the water-soluble molecules out of your clothes. But the soap can also take the lipid-soluble molecules out of your clothes. And that is why we clean our clothes with a detergent, so that they will become cleaner than if we were only washing it with water alone. And actually, if you were washing the clothes in lipid, which we don't, if you were to do that and you were to add the detergent, it would be cleaner than just washing it in lipid alone also, because it would remove the water-soluble agents as well as the lipid-soluble agents. Any question about that? So phospholipids are biphasic. They can dissolve in both lipids and water. When we put them in, meaning phospholipid molecules, in water, they will line up like this. Let me blow that up a little. And that's because the phosphate portion is the portion that likes to be around water, and so it'll come this way facing the water and that way facing the water. And then the fatty acid portions don't like to be around water, so they will come together. Okay? And in the next slide we'll see that it doesn't just stay linear. The phospholipids will actually form a liposome or a micelle in water. And the point of the liposome and the micelle is for the fatty acid portions to be away from the water, which are over here, and they can also be inside the liposome here. And this liposome should remind you of something. What does this remind you of? So the fatty acids, when you put them in water, they form this structure. And what does this structure remind you of? Come on, somebody's got to guess.
A cell?
Yeah, it reminds you of a cell. There will be water on the outside of a cell. So let's talk about a red blood cell. It'll have blood on the outside, and that's mostly water. And on the inside of the cell is cytoplasm, and that's mostly water. And then right here is the lipid bilayer of the cell membrane, and it'll have the phosphate group facing out, facing the water, and the phosphate head facing in, facing the water of the cytoplasm, and then the fatty acid portions of the phospholipid will be on the inside, making the internal part of the cell membrane. And the cell membrane actually is mostly phospholipids in a structure like this, with the phosphate heads facing the cytoplasm, the phosphate heads facing the outside of the cell, and then the fatty acids being inside to make the lipid bilayer. There are other molecules, of course, in a cell membrane, but the number one molecule is a phospholipid. Any question about any of that? All right. The last group of lipids we want to talk about are steroids. And if you have an OH group on a steroid, we call that a sterol. Steroids are four connected rigid carbon rings that are characteristic of this structure, this group. And the sterol you're probably most familiar with is cholesterol, which is this structure with the OH group on it. We have cholesterol in animal plasma membranes. So in our plasma membranes, we have cholesterol in the cell membrane, and the cholesterol adds strength to the cell membrane. Remember, animal cells do not have a cell wall, and all they have is a cell membrane, and we have this cholesterol in the cell membrane to give strength to the cell membrane. Any question about any of that? Steroids are the one lipid which is not made up of a fatty acid and a glycerol. Of course, a phospholipid also has a phospho group to it, but lipids are typically made up of glycerol and fatty acid. And we can call these monomers of the fatty acid. So most lipids are made up of monomers, but not the steroids and the sterols. They are one molecule like this, and there is no monomer to them. Any question about any of that? All right. Where do you find lipids in the food that you eat? Not lipids, sorry. Where do you find cholesterol in the food that you eat?
Meat.
Say again?
Some meat.
Yeah, in meat. You find cholesterol only in animal products. It can be dairy products as well as meat, and I suppose eggs, which I don't know if that's dairy or not, but you only find cholesterol in animal products. This molecule is not found in plant products. So if you wanted to have a low cholesterol diet, you should eat lots of fruits and vegetables and avoid dairy products and red meat. To reduce your cholesterol, you can eat lean meat, and you can have low-fat dairy products. Do they have non-fat dairy products? I think they do, yeah, like skim milk would not have any cholesterol. All right, that's it for lipids. Any question about lipids? If not, let's move on to the third of the biological molecule, the proteins. Proteins are essential in cell structure and function. Proteins may be structural proteins found in bone, muscle, and hair. They may be enzymes which make enzymatic reactions happen faster. Proteins may be transport proteins that move chemicals across the cell membrane and either into or out of the cell. Proteins can be regulatory proteins which regulate body functions. For example, hormones, like a growth hormone in a child which is growing. Proteins can be responsible for movement. The reason why we're able to walk is because we have proteins to make our muscles move. So muscles are largely protein. But it's also true for the microbes. If they're moving, it's because they have the protein. I'm talking about the bacteria microbes. It's because they have the proteins for making a flagella. Proteins are also responsible for some of the bacterial toxins. Most of the exotoxins are proteins. But proteins are also antibodies. And these are proteins that our body makes to defend ourselves against infection. So as you can see, proteins are playing a number of different roles in an organism. And proteins are probably the most diverse of all of the biological molecules. The reason why proteins are more diverse than the other biological molecule is that proteins are made up of 20 different monomers, whereas lipids only have a few monomers. Carbohydrates have a couple of different sugar monomers, and the nucleic acids only have 4 monomers. Well, proteins have 20 different monomers that we call amino acids. Let's take a look at the general shape of an amino acid. An amino acid always has a central carbon, and it's always bound to a hydrogen. On one side of the amino acid, we have an amino group, and that's the NH2 group shown in blue. And on the other side of the amino acid, you have the carboxyl group, the COOH group, shown in pinkish color. And then carbon binds to a 4th element, and the amino acid will always bind to an R group. The R group can vary, and we just put an R there to mean that this can vary. Each different amino acid will have a different R group. One R group is this group shown for the amino acid tyrosine. And each amino acid has a different R group, so this R group determines what amino acid it is. Any question about that? You should know the general structure of an amino acid. You do not need to memorize the next 2 tables, but I just want to show you that there are 20 different R groups, at least in human, and it's, let me blow that up a little. It's colored a darker shade of blue here. The simplest R group is in the amino acid glycine, where the R is just hydrogen. But each of the different R groups will vary for each different amino acid. Some of the R groups, actually all of these R groups here, are not, what's the word I'm looking for? Are not hydrophilic, meaning they are hydrophobic. So these R groups are all hydrophobic. They do not like to be around water, including that one. Some of the R groups have portions of them which are hydrophilic, like this OH group here, and these 2 NH groups are going to like to be around water. And so part of this R group is hydrophilic. Now this region of this R group is not hydrophilic, and this region would be hydrophobic. It's important to realize that some amino acids have R groups which are hydrophobic, and others have R groups which are hydrophilic. All amino acids have hydrophilic regions because the amino group and the carboxyl group are always hydrophilic. So proteins are both hydrophilic and hydrophobic. Any question about that? The hydrophobic will be certain regions of the R groups, and then the hydrophilic will be the amino group and the carboxyl group, and maybe some of the R groups. Any questions about that? All right, let's talk about amino acids and how they get together to make the proteins. Amino acids are linked together by a peptide bond. A peptide bond is an unusual bond that is formed by dehydration synthesis, meaning OH comes off of this amino acid and hydrogen comes off of this amino acid to make water, and then the nitrogen here binds directly to the carbon there. And that's an unusual bond, the nitrogen into carbon. You don't find it in very many places in an organism or in the universe in general. Nitrogen binding to carbon doesn't happen all that often. And so this bond, we call it a peptide bond because it does happen in peptides. Peptides are sort of like a short protein, meaning an unfinished protein that's being built is a peptide. Now, what am I supposed to say about a polypeptide chain? Oh, you can link the different amino acids together to form a polymer that we call a polypeptide. And it'll be a chain of the different amino acids hooked together. When you're building a protein, you will be adding amino acids, and as you're adding amino acids, strictly speaking, it is a polypeptide. Once you've added the last amino acid to the growing protein, we can then call it a protein. So that's the only difference between a polypeptide and a protein. If it's all finished growing, meaning all finished adding amino acids to it, that's when we call it a protein. If it's any size smaller than the final protein, we call it a polypeptide. So a polypeptide is just a chain of many amino acids linked together, and when all of them are added, that's when we call it a protein. Any question about any of that? All right. Proteins have 4 levels to their structure. And let's see if this link works and I can get it to open. And then we'll watch a little video discussing the 4 levels of protein structure.
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now, these 2 amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the 2 amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. we have one hydrogen left over. We lost two once we lost water. So this is a dipeptide because we have two amino acids combined. And so this amide bond that we see here, that is the peptide bond. We formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein, and there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid, and each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we can see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
Okay, any questions about that video? Everybody got it?
Yes.
Okay, somebody actually answered. That's good. Oh, where am I? There we go. So let's talk a little bit more about the primary structure, the secondary structure, and the quaternary structure. The primary structure is what happens when the protein is just made. The primary structure is just the sequence of the amino acids, one amino acid after the other. Once the protein is made, these amino acids can start interacting with each other, and that gives rise to the secondary structure. The secondary structure is localized interactions between neighboring amino acids. So the secondary structure is only the interaction of nearby amino acids. Like in this one, it looks like we have a hydrogen bond between this amino acid and that amino acid. We could have repulsion between this amino acid and that amino acid. Repulsion could be possibly when one amino acid has a positive charge and the other amino acid has a positive charge. The attraction could be when one amino acid is a negative charge and one is a positive charge, or the attraction could be when one is a polar group and it forms a hydrogen bond to another polar group in the amino acid. There's one other attraction which I almost hesitate to talk about, and that is lipids like being around other lipids. And what they do is they have van der Waals forces where they like to be together. And that's a weak attraction, but, I don't know, maybe these two could be, have van der Waals forces attracting them together. Is that clear? You don't really need to know much about that one, but if you remember from your preliminary course, you probably discussed van der Waals forces, and it's the things that, like when you put oil together, the oil wants to stay together, and that's van der Waals forces keeping the oil molecules together. Okay? Any question about any of that? Those are the different attractive forces that can form the secondary structure. Okay? The video only talked about one, I think the hydrogen bond, but in reality there's also attraction between ionic groups, repulsion between ionic groups. Oh, and I forgot to mention, there can be repulsion, like right here, if they're both polar group. Is that right? No, but they're both, if one is a polar group, I guess it'd have to be this one. One is a polar group and one is a non-polar group. They will be repulsed from each other. Okay? So you can have various forces of attraction and various forces of repulsion to form the secondary structure. What is key in the secondary structure is that it's only the localized nearby amino acids which are interacting with each other. All right? And that gives rise to different shapes, which I'm not going to hold you to. There's the alpha helix, which is shown here, the beta pleated sheet, and then the random coil, which we're not going to talk about those. Okay? Now, the tertiary structure has the same attractive forces as the secondary structure, meaning there can be hydrogen bonds forming, they can be hydrophobic interaction between two amino acids which are both hydrophobic, and so they'll want to be together. There can be repulsion between one that is hydrophobic and one that's hydrophilic. There can be repulsion between, if this was positive charge and that's positive charge, that'll be repulsion. There can be attraction. If this is positive charge and that's negative charge, that'll be an attractive force. So all of that can happen in the secondary structure and in the third structure, the tertiary structure. But there is one other attractive force that you can find in the tertiary structure, and it's not found anywhere else. And that is the disulfide bridge. Let me blow this up so you can see that better. The amino acid cysteine has a sulfhydryl group on it. And let me end this now. And I got to open Word to show you this. So we can have the amino acid cysteine, that's the abbreviation, and it's having a sulfhydryl group on it. Well, it can get together with another sulfhydryl group on another amino acid cysteine, okay? And the hydrogen can come off, so let me erase it, and then this hydrogen can come off, and then the sulfurs can be connected with a polar covalent bond. This is a very strong bond linking this cysteine with that cysteine. how to show you how strong that bond is. My mother used to curl her hair. She would get a curling iron and put curls in her hair and put a bobby pin so that the hair would curl, and then she would put the curling cap on and then turn on the machine, and it would put moisture and heat in the curling thing, and then would warm up her head and make it moist, and then would form curls in her hair. And then she would go out in the moist Oregon air where it might be misting, and the mist would make her curls sag. And the reason is that the moisture in the air could hydrogen bond with the curls, which are formed by hydrogen bonding, and it's a very weak bond. And so the curl would only last, I don't know, a couple of hours, maybe a day at most if you stayed away from moisture. And if you got in the moist air, like if it is misting, the curl would unfold. So it was a weak link. However, she never did this, but if she were to go in and get her hair permed, what they do to perm your hair is they form a disulfide bridge in your hair between two cysteine amino acids. And this is a very strong bond, and as you know, you can perm your hair and then take a shower, and your hair is still permed. In fact, your hair will be permed for about a month of taking the shower. And that's because this bond is very strong. Anyways, the disulfide bond can form in the tertiary structure of a protein, and it doesn't form anywhere else. The other thing that is different about the tertiary structure is that the tertiary structure can have amino acids interacting, like right here, it's being attracted to amino acid here, but it's not the neighboring amino acids. In the secondary structure, only the neighboring amino acids were interacting with each other. In the tertiary structure, the amino acids can act all over the protein. In fact, this one is an amino acid at the end of the protein interacting with an amino acid near the start of the protein. And that's the tertiary structure, where the protein amino acids start interacting, and then the protein takes on its shape. Is there anything else I want to talk about that? And the amino acids can interact, either be repulsed, what would be an example of that? Maybe a repulsion between this amino acid and that one. And then there will be an attraction, let's say, between this amino acid and that amino acid. Okay? And the attraction and the repulsion can be the same as in the secondary structure, with the exception you can also have the attraction between the two cysteamine amino acids, where they can form a disulfide bridge. Okay? Any question about the tertiary level of a protein structure? I'm not hearing anything, so I'm going to go on. For many proteins, the tertiary level is the last level that they will have. Like most enzymes only have a tertiary structure to the protein. But some proteins have a quaternary level of the protein. And these are proteins which are made up of more than one polypeptide unit, like hemoglobin, the antibodies, and the insulin. They're made up of more than one protein. And these different, I shouldn't say protein units, I should say, these different protein units have to come together and have attraction and repulsion between them to make the final protein. and that will be the quaternary structure. When a protein consists of 2 or more polypeptides. So only some proteins have a quaternary structure. All right, any questions about that? So those are the 4 levels of the structure of a protein. Proteins can have a globular shape, and these are overall roughly spherical proteins. They're compact proteins, and they can be an example like hemoglobin or insulin. Most enzymes are globular proteins. You can also have a fibrous protein, and they're thread-like, and that would be like collagen. Okay? Any question about that? Proteins can also make combinations with other molecules, and these we call conjugated proteins, where the amino acids bind to another molecule, either organic or inorganic. The name of the molecule usually comes from their non-protein component, like glycoproteins, or when a protein binds to a carbohydrate. A lipoprotein is when a protein binds to a lipid. Any question about that? And we can find lipoproteins in your blood, glycoproteins also. Glycoproteins can also be found on cells, like the surface of the cells. Proteins can denature. For a protein to have its correct shape, all of the elements have to get together, and then the protein has to bind in its correct shape. And it has attraction and repulsion in the protein for it to take on its correct shape. You can denature a protein by making the protein take on a different shape. That can break hydrogen bonds where they're supposed to be. It can form hydrogen bonds where they're not supposed to be. Proteins that have lost their shape and no longer function, we call them denatured proteins. And you can denature a protein by putting it in the wrong temperature, putting it in the wrong pH, or putting it in the wrong salt concentration. An example of a denatured protein, when you get your egg white and you crack the egg and put it into the pan, that is the normal protein. And then when you heat the protein of the egg white and it turns white and solid and then you eat it, that is denatured protein in the egg white. And that's denatured by the temperature. Any question about any of that? All right, why did that come up? All right, any question about proteins? Everybody's good. So let's move on to our 4th biological molecule, nucleic acids. There are 2 main nucleic acids, DNA and RNA. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. All of these consist of monomers that we call nucleotides. A nucleotide is a molecule that has a sugar moiety in it, and it's a 5-carbon pentose sugar. And for RNA, it's ribose. For DNA, it's deoxyribose. There will also be a phosphate group attached to the sugar moiety, and then a nitrogen-containing base attached to the sugar moiety. In DNA, it's the nitrogen-containing base which binds to the other strand of DNA. So one strand of DNA, the nitrogen-containing base, hydrogen bonds to another nitrogen-containing base in the second strand of DNA, which we're sort of seeing a little bit of that one here. Any question about that? All right, so that's the nucleotide, and the nucleotides make up the DNA and the RNA, and all the DNA and the RNA are strings of nucleotides put together. I've already talked about that. There are two types of nitrogen-containing bases. There are purines and there are pyrimidines. For DNA, the purines are adenine or A, or guanine or G. And I don't care how you remember that, but you should know that you should know the names adenine or A or guanine or G for the nitrogen-containing base of the nucleotide. For the pyrimidines of DNA, we have cytosine and thymine. And they have a different shape. The purines are a bigger molecule, and the pyrimidines are a smaller molecule. How you can remember them, at least for DNA, is pyrimidine has a Y in the name. And of the nucleotides which are pyrimidines of DNA, they have a Y in the name. So cytosine and thymine have a Y in the name. Okay? Now, for RNA, we have A, G, and C, but we do not have thymine or T. In RNA, we replace thymine with uracil, which is a pyrimidine, and you're just going to have to remember that in RNA, you replace the thymine, which is a pyrimidine because it has a Y in it, and replace that with uracil, which is a pyrimidine, but it doesn't have a Y in it. That's why I said you can remember it for DNA. For RNA, you have to just remember the trick that A, G, and C are the same nucleotides in RNA. And then the thymine isn't in RNA, it's replaced with uracil, which is a pyrimidine. Okay? Any question about any of that? Everybody seems to be good, or at least they're not talking. The bases bind to each other via hydrogen bonding, and they only bind to their complementary base. So an A will only bind to T in DNA, and a T will only bind to A. Now, we're talking about DNA. If we have RNA, you don't have T, you have U, and I'm just going to leave it there. And then C can only bind to G, and G can only hydrogen bind to C. So those are the complementary bases. You should also know that the A and the T binding have two hydrogen bonds in them, and the G and the C, do I have that anywhere? The G and the C have three hydrogen bonds between them. So the C and the G, it's a little stronger bond than between the A and the T, just because there's one more pair of hydrogen bonds. All right, there we got the G and the C having three hydrogen bonds. Any question about any of that? All right. DNA is held together by the sugar phosphate backbone in one strand. So one strand of DNA, let's use this one, it's held together not by the nucleotide bases. We're talking about one strand of DNA. There's the sugar right here, which is linked to the phosphate right here, which is linked to the sugar. And so it's the sugar phosphate backbone which holds one strand of DNA together. Now the double strand is held together by hydrogen bonds between the nucleotides of this base and the nucleotide of that base, and the nucleotides do not hold the one strand of DNA together. They hydrogen bond to hold the two strands of DNA together. And DNA is twisted sort of like a spiral staircase, and the nitrogen-containing bases are the steps on the stairwell. And I guess the sugar phosphate moiety are what's holding the stairwell together. Any question about any of that? All right. In DNA, the sugar is deoxyribose, shown, well, that's not shown there. Shown here, right there, deoxyribose. In RNA, we have the sugar ribose. It has one more oxygen added, so there's two OH groups on it. In the DNA, there's only one OH group on it. RNA is held together by the sugar phosphate backbone, and RNA is only a single-stranded molecule, so it is only held together by the sugar phosphate backbone, which is a covalent bond. I've already mentioned that they have a different base from T. They have U instead of T, but the other three bases are the same. So A, G, and C are the same. There are three major kinds of RNA. There's tRNA, or transfer RNA, mRNA, or messenger RNA, and rRNA, or ribosomal RNA. The different types of RNA are important in protein translation. Okay, any question about any of that? All right, so that's it for the nucleic acids. Let's talk a little bit about adenosine triphosphate, because it's an important molecule, and if the cell needs energy, it usually obtains that energy from ATP, meaning ATP is the primary energy source for most cellular activities. We call ATP the energy currency in cells. Currency is something we use to buy things like money, and whenever the cell needs energy, it usually takes that energy from ATP. ATP has a sugar and a nucleotide, which is of course adenine, and then three phosphate groups. This phosphate group with the squiggly line is a high energy bond, and so is that phosphate group of the second phosphate. And if the cell needs energy, it will break that bond right there, and that will release a lot of energy which the cell can then use. If the cell needs more energy, it can also break this phosphate group right there. Okay? Any question about any of that? So ATP is made by dehydration synthesis. We take ADP, adenosine diphosphate, shown here, and we add phosphate. It's just an inorganic phosphate. And then you need energy to make this reaction go, and that'll form water and ATP, adenosine triphosphate. You can break ATP by breaking that bond by adding water to form adenosine diphosphate, ADP, and then phosphate will come off, and that will release a large amount of energy. And that's how the cell uses ATP to get its energy needs. All right, any question about any of that? So here's a question for you about hydrogen bonds. What molecules have we discussed that have hydrogen bonds to give the molecule important properties? What molecules have we discussed in this lesson that have hydrogen bonds that give the molecule important properties?
Water.
Yeah, definitely water. You wouldn't have liquid water without liquid water forming hydrogen bonds to another water molecule. What else? What other molecules have we discussed?
Proteins.
Say again?
Proteins.
Yes, proteins form hydrogen bonds, and that's important for the protein to have its shape and fold properly. There's one we just talked about, right here. What's holding the DNA together? The two strands. Come on, people. What is holding the two strands of DNA together?
Hydrogen bonds.
Yeah, hydrogen bonds from one nucleotide are bonding to the complementary nucleotide. So DNA also has hydrogen bonds. And what would happen to these molecules if all the hydrogen bonds were to disappear? This isn't that difficult of a question. You wouldn't have double-stranded DNA if all the hydrogen bonds were to disappear. We wouldn't have liquid water if all the hydrogen bonds would disappear. If all the hydrogen bonds were to disappear in liquid water, the water molecules would all evaporate and become gaseous water. In proteins, if you didn't have the hydrogen bonds that are supposed to be there, the protein would unfold and take on the wrong folding pattern, and the protein probably would not function. It certainly wouldn't function at its optimal level. All right. Any questions about Chapter 2? If not, that's it for Chapter 2. We can close that. Let's begin Chapter 3. So, as usual, I will give you a first slide which will introduce the major goals and give you a rough outline of what we'll be doing. So, once again, know the terms, know what is resolution, refraction, what are stains, know what a basic and acidic dye is, know what a bacterial smear preparation is, know what a mordant is, know what peptidoglycan is. Understand the sizes of organisms and the properties of microscopes. Be able to understand the major differences between the light microscope and an electron microscope. And then understand why staining improves resolution. Know that there are three types of stains: a simple stain, a differential stain, and special stains. And then lastly, be able to understand the Gram stain, its procedure, and the basis for its differential staining, meaning why does it give differential staining. Any questions about any of that?
I'm sorry, before you start, did you want us to let you know about 6?
I'm sorry, I didn't hear that.
Did you want us to stop at 6?
Is it time to begin the lab? Is that what you're telling me?
Yeah.
Okay.
Is it?
Yeah, we should probably end now or shortly so that I'll have 20 minutes to record this. All right. Are there any questions? Boy, we didn't get very far at all in chapter 3. We're probably going to fall behind schedule. All right. I will see you at 6:30 for the lab. Okay?
Thank you.
All right.
Thank you.
Thank you.
All right.